Welcome back, everyone. Time to continue with our program. We now have a session of 9 more pitch presentations, 5-minute pitch presentations at the pre-doc level, as we call it. After this, you've heard them all 18, so don't forget to vote. Then it's on one of the forms you will find in your program booklet. And hand in the forms when you leave the auditorium later when we have our lunch break. During the lunch break, don't forget to start looking at the posters. There are 60 posters in competition, and we also expect you to vote for the best poster. Best poster award is usually divided between best posters selected by the public and also best poster selected by professional jury. So we have 2 prizes, but one of them depends on you. So I now give the floor to the presenters, and the first one is Christine Rund of Oceans and Lakes. The floor is yours.
Hi, my name is Christine Rund, and I'm presenting to you first results of my ongoing master thesis research. The ocean covers 70% of the Earth's surface. It is the largest ecosystem on Earth, and it provides essential services to us like food, like recreation or the oxygen we breathe, and also provides economic benefits. The Sustainable Development Agenda, which was adopted by the UN General Assembly in 2015, includes the sustainable development of the environment and also puts the ocean in the wider sustainable development context for the first time, namely in terms of SDG 14. But despite the recognition of the importance of the ocean for the whole agenda in achieving the SDGs, it still ranks low on agendas of policymakers and also receives quite low funding. So can we conclude from this, below the sea, below the radar, and how can the SDGs help us in raising awareness of the ocean? In order to investigate this, I conducted an online survey with the alumni of my master program, so with people educated in marine sciences from all over the world. And I asked them on their perception of the importance of SDGs, and you can see that now SDG 14 ranks highest. But if I compare that to other surveys, what I find highest most is decent work and economic growth, which is rather in the middle, or quality education, which is also ranked high in my survey. But good health and well-being and also no poverty is rather in the middle of priorities of ocean-educated people. Zero hunger is even lower. So what we can conclude from this is that education has an impact on the perception of SDGs. Furthermore, I asked them about how can we realize the goals. And they said mostly that the local level is really important. However, also we have to include all different levels, so individual, local, national, and global. Limitations for reaching the SDGs, in their view, are mostly short-term decision-making, but also different priorities. And I just go on. And for a second part of my survey, I then wanted to look at what we can actually do to raise the awareness. So I wanted to take it a step further. And I conducted an expert survey with an interdisciplinary group of experts, and we developed 2 projects that have the opportunity to raise awareness. And we evaluated them in terms of the impact, the likelihood of implementation, and its feasibility in terms of cost, effort, and available capacity. The first project is building on an existing citizen science project here in Belgium, Sea-Watch, where people or citizens measuring the beaches on regular basis. And we want to add on top of this a kind of soft GIS. So how do people feel when they are on the beach? So it combines environmental science and social sciences on a local scale in Belgium, combining different SDGs. SDG 14 on the ocean, SDG 15 on the land, and SDG 3 on good health and well-being. The second project is an ocean footprint calculator that transforms the personal ecological footprint to ocean impact. And it might be also possible to trace behavioral changes if you apply this over a longer period of time. And this can be applied very locally, so in schools for citizens, kids, but also in a global scope involving fishing industry maybe or the cruise sector, and so might have a higher impact than the first more local project. Here we combine SDG 14 on the ocean, 3 on climate action, and 4 on education. My preliminary results, so if I come back to the question I raised in the beginning, below the sea, below the radar, yes, in many cases it is this way, but we can also see that education matters, that education is a way of raising awareness, and that a workshop can be a useful tool to develop projects that link different SDGs to have a higher impact, and also that citizen science might be important in this area. Thank you for your attention.
Earth is a coastal planet. It has 1.6 million kilometers of coastlines. That's about 40 times around the equator. Did you know that 83% of all countries have coastlines? Coastlines in different shapes and forms, with different climates and ecosystems. And people like you and me, we are attracted to coasts, especially sandy coasts. Perhaps we like it a bit too much. We like it so much that it has implications for coastal evolution, sometimes with unbalanced benefits. Currently, the coastal population is growing faster than the inland population, and they face increasing challenges such as sea level rise, coastal erosion, and an increased risk of flooding. Coastal dunes, they are one of the most efficient natural barrier systems against flooding. Opposed to sea dikes, they are built by nature, and the disadvantage is, however, that they are very dynamic, and the safety level provided by them varies in time. And here is where my research begins. So my name is Glenn Stripsteen. I'm a PhD student at KU Leuven campus Bruges, and I study the wind-blown sand transport at the Belgian coast. And in today's talk, I'm going to share with you how the Belgian dunes are behaving. So as you all noticed in the past few days, there is a storm raging through Belgium with wind speeds up to 115 kilometers an hour. Those high wind speeds caused a lot of sand transport on the beach. Wind-blown sand transport is a topic that has been investigated by scientists since the beginning of last century, but it's only during the recent years that it has been investigated in coastal areas. Research about how are dunes growing due to those processes is of course very relevant, and often people have no idea how these processes really work. So the Belgian coast, stretching between the borders of France and the Netherlands, is characterized by sandy beaches. It's also divided by 255 coastal sections, defined by unchanging boundaries. And that's also used in my study. So maybe you don't know it, but half of the Belgian coast is protected by coastal dunes, and they range in height between 5 and 30 meters above sea level. And since the 70s, the Belgian government has been monitoring our coastline by taking images from the air, and since 1990 by taking airborne laser data to see how our coast is developing. So from those data we can make elevation plots, difference plots, and we can determine how the coast is changing. But how do our dunes change now? Well, these graphs show dune volume in time for different coastal sections along the Belgian coast. Sure, the dune volume is changing over time, but eventually we can see where it is moving. Some dunes are growing in time, so some dunes are eroding in time. And fitting linear trend lines for all the sections can see if this linear trend is valid for the entire Belgian coast. And we have found that 88% of all the Belgian dunes have a linear model in time. They are represented by a linear model in time. It is observed that for the entire Belgian coast, most dunes are growing in time, and some dunes are eroding in time. We have calculated an average dune growth of 6.2 cubic meters per meter per year, with some longshore variability. You can notice between the Haan and Wendeunen that we have there the most dune growth, and that is attributed by the creation of a white beach due to nourishments in the 90s. The dune regions next to France and the Netherlands have a slower dune rate, and some regions do not grow linear in time. For example, the dune region in the Banne, that is 1 kilometer long, does not grow linear in time because they implemented a concrete wall in front of the dune to prevent it from eroding. That was one of the protection measures in the 80s. And that brings me to the end of my talk. My name was Glenn Stripsteen, and my taking home message for all of you is that the dunes at the Belgian coast are growing. Thank you.
Hi everyone, good afternoon. My name is Inge. I am a PhD student and I will tell you a little bit about part of my PhD thesis that is incorporated into a project called PCAT for cod, and it looks at the effects of seismic survey on the behavior and movement of cod. So first of all, what is a seismic survey and why should we care about the effects of this on fish? When in search for offshore sources of fossil fuel, companies use a vessel that pulls an air gun behind them, which is called the seismic source, and this air gun emits a sound blast towards the seabed. This air gun source is dragged behind the vessel, and behind that there is an array of hydrophones which picks up the reflection from the seabed. And using that, images can be formed of what is below our seabed. The sound that is emitted by the air guns is very low in frequency and can be heard by many marine fish, including cod. So how do we track an animal that's in the sea? Well, GPS signal doesn't travel through salt water, so we use something else which is called telemetry. With this technique, a tag is inserted into the fish, which emits a signal, and with this signal, we get information on the presence, the position, and sensor data, which in our case includes information on the depth of the animal through a pressure sensor and the overall acceleration of the animal through an acceleration sensor. And where do we study the behavior of cod? Well, previous studies have shown that cod reside in areas where there's structure, which is like an offshore wind park, and they spend prolonged periods of time there. So what we did is put a bunch of hydrophones that can receive the tag signal in this area, all the white dots you can see here, and we tagged 57 fish around this area. So we got data on their presence, position, and depth and acceleration use, while there was also a seismic survey going on in the area, which approached our study area up to 2 kilometers. And what does this data look like? Well, these are some first plots of what we got out of this data. Here you see the overall presence of the animals and the time on the x-axis. And the red lines indicate the period when the seismic survey took place, so when we had the sound exposure. And you see that over time there is a decreasing detection of our animals, but you don't see that there's straight away something happening within the seismic period. And when we zoom into the individual fish, so every line is a fish and every dot is when it was detected in the area, you see again that there's no immediate sign to think all the fish leave the area when the survey is taking place between the red lines. We also get information on the movement of the animals. So here you see per plot it's one zoomed into one turbine with receiver stations around that, and the position of our fish within this system. And there's a lot of change in aerial use of individuals, and this is very much bound to individuals. For example, in the lower ones, you see the blue fishes using a different area than the purple fish. But again, you don't know what that could mean yet. As well as this is what the information on acceleration looked like. So you got from 0 is they're not accelerating at that point, up to 5 when they're accelerating quite a lot, 5 meters per second. And you see that there is fluctuation in the overall acceleration through time. It looks like there could be some indication for less acceleration at the onset of the survey, right after the first red line. But you also see a definite pattern in this data, and when you overlay day and night patterns, this kind of fits that pattern well. So the gray bars are nighttime, and there's more acceleration then. So what we're going to do now is we're going to perform some further analysis on this data to get information on the spatial use of the animals, as well as different behaviors they might be displaying. And we're going to correlate that to the acoustic data, so information from our hydrophones in the area, as well as the position of the vessel at any certain time. And I hope to be able to show you some of the results later on. Thanks very much for listening. If you have any questions, come by later. And there's a nice flip stand there which explains telemetry, so if you have questions on that, go and see them. Thanks.
Good morning, everyone. I'm Annelore van Nieuwenhove from the Marine Lab of the VUB. Due to the increasing market demand for cephalopods worldwide, many traditional octopus fisheries in the Indian Ocean have expanded very fast in the recent decades and have shifted their focus from local and inland markets to international export. Just as in other regions worldwide, the landings of these octopus fisheries are decreasing and concern over sustainability is raised. To maintain the sustainability of these socio-economic important octopus fisheries, management actions must be taken, such as the establishment of marine protected areas. Insight into connectivity is vital for a correct spacing of marine protected areas. When populations are connected, it means that there is gene flow among those populations or an exchange of individuals among geographically separated subpopulations. Because most of the reef-associated organisms are not very mobile during their adult life stage, connectivity is essentially larval dispersal from one reef to another. When populations are not connected and they don't receive an input of such foreign individuals, local overexploitation might lead to extinction of that population, which can of course be a catastrophe for local communities that rely on fisheries. Because pelagic larvae are extremely difficult to track in the open ocean, population genetics is a useful technique to study connectivity and gene flow patterns. So in order to obtain baseline data for management plans, the present study investigated the diversity of Octopodidae in the Western Indian Ocean and the connectivity among populations of two commercially imported species, Octopus cyanea and Octopus vulgaris. So how did we do this? We collected arm tips of octopus on landing sites and fish markets at 15 different sites in Tanzania, Kenya, and Madagascar. From those arm tips, the DNA was extracted and a fragment of the mitochondrial cytochrome C oxidase subunit 1 gene, also known as CO1 gene, was amplified and sequenced. CO1 gene is also known as a barcoding gene since it allows scientists to identify the species by comparing the sequence with sequences of many different species in a sequence database. And this results then in an identification summary as output. It's the same principle as scanning the barcode on the product of the grocery store. It's very simple. So besides our own collected tissue samples, we also used 41 CO1 sequences of Octopus vulgaris from different localities worldwide, which are represented by the yellow dots, and these were available on online databases. So besides telling us something about the species, CO1 can also give information on connectivity. How does it work? The more similar the sequences of individuals from different populations are, as you can see on this picture, the higher the gene flow or the connectivity. When the sequences are more different, we say there is high genetic differentiation and therefore low gene flow and low connectivity. So DNA barcoding revealed the presence of 5 different species within our samples, with first records of Callictoctopus luteus and Octopus oliveri in the Western Indian Ocean. For Octopus cyanea, significant genetic differentiation was found among Kanamai, southern Kenya, Tanzania, North and West Madagascar, Southwest Madagascar, and East Madagascar. For Octopus vulgaris, limited connectivity was found among populations from Brazil, populations from Madagascar, and then all the other ones were grouped as one. So what are the take-home messages? There is a higher species diversity of Octopodidae in the Western Indian Ocean than previously thought. For Octopus cyanea, we recommend, based on our results, 4 separate management units in the Western Indian Ocean. And for Octopus vulgaris, populations from Brazil and Madagascar were so genetically differentiated that they might be evolving towards speciation and therefore should be considered as separate species in FAO fishery statistics. Of course, the present study was a bit more complex than the way I presented it here, so people that want more in-depth information on the genetic part or statistical analysis can always talk to me during the breaks. Thank you very much.
Okay, hello. I must admit something, talking to 400 people, kind of stressful for me, for everyone who's presenting here. So let's see what this stress does to my body. So all you people are staring at me, the clock is ticking, and this sends a signal to my brain. And my hypothalamus gets active, sends a signal to my pituitary gland, which sends on its turn signal to my adrenal glands. And these adrenal glands will produce cortisol. Cortisol is a known stress hormone in the humans, and it will travel through my blood and it will have effect on different organs. So, for instance, my heart is beating really fast now due to the cortisol. My brain maybe have a blackout due to the cortisol. So all different effects of cortisol. So we are here to study marine species. So the flatfish, common death in this case, has exactly the same hormonal axis that causes the production of cortisol. Two differences. First of all, I assume that the flatfish will not be stressed by talking to 400 people. And secondly, the outcome, so the cortisol gets produced by the interrenal cells. These are exactly the same cells as in our adrenal glands, except they are not organized in glands. So exactly the same happens in a flatfish and so a fish swims in the water, something happens and the fish gets stressed. So what happens to the cortisol levels? We have the normal daily variation in the cortisol levels. Something happens, stress, and the cortisol levels will increase. So this is called acute stress. Normally, the fish will adapt to it, they will cope with the stress, and the cortisol levels will lower again. But if the stressor, for instance pollution, remains for a longer period of time, it doesn't cause acute stress, but it causes chronic stress. So in that case, the cortisol levels remain high, and this can cause maladaptive effects for the fish. So how can we measure this cortisol? How can we measure stress? So we have first, the acute stress phase can be measured by analyzing the blood of the fish. But the second phase, chronic phase, can be measured by, can be pinpointed by measuring the cortisol in the scales. Because the scales will capture cortisol that goes through the blood, and they will capture it and keep it in the scales. So you have a whole life history of stress of a certain fish in the scale. So this method was pinpointed for different aquaculture species, but not yet for wild-caught marine flatfish. So we thought, okay, this will be the same in all fish, so no problem. So we sampled a lot of common death. We did the analysis, but all the cortisol levels were really, really low. So this was not exactly what we expected. So we started asking questions. So can the flatfish, like any other aquaculture fish, capture this cortisol in the blood and store it in the scales? Or maybe they have a difference. So we set up an experiment. We have 3 groups, a wild-caught fish, sampled right after catch. And we have a control group. They were caught and then kept in an aquarium. And then a cortisol group. They were caught, kept in an aquarium, and got feed that was enriched with cortisol. So what we see is this: if we analyze the scales, we have a clear difference in cortisol level in different scales. So the wild fish have low levels of cortisol, and the cortisol fat group has high levels. So this confirms our hypothesis. Common depth is not different than any other fish. Common depth is able to store this cortisol in the scales. So this means that this technique of analyzing cortisol and analyzing the scales can be used as a biomarker. So we can see if something happens in the environment, we can analyze the scales, and we can see if something, if the fish are stressed or not, if it's a stressed environment or not. Of course, we are still at the start of our investigation on this topic, so we still have a lot of work to do to see if really stressed fish, or if we expose them to some pollution or something, if these levels rise or not. So we have a long way to go, but it's already very promising. Thank you.
Good morning. I'm Helena Voets, a PhD student at the Royal Belgian Institute for Natural Sciences. I've recently completed a series of experiments on blue mussel, a key species in offshore wind farm falling fauna and aquaculture, and I'm showing it's adversely affected by a changing marine climate. This work has been done within the framework of the Persuade project, and in this project we're looking at the combined local and global pressures acting on our marine environment. The objective of this project is to investigate the impact of these combined pressures on food web interactions and on nutrient cycling of the coastal ecosystem, and integrate these dynamics over the water column and the sediment. Based on the dominant communities of falling fauna and the vertical zonation on the offshore wind farms, we've selected 3 model species. In this presentation, I'll present the work done on Mytilus edelis, the blue mussel, taking into consideration its aquacultural value and its potential of the shell biofilm. A fully cross-lab experiment was set up at Ghent University, and 900 mussels were harvested at the North Sea aquaculture pilot projects, and these were then randomly distributed over 4 environmental treatments. We have a control treatment, an ocean warming treatment, ocean acidification, and then both pressures were combined in a climate change treatment. The latest series of these experiments ran over the summer of 2018, and over the course of 6 weeks, the temperature and pH was manipulated with an EKS system. During the 6 weeks, survival and condition were monitored closely, and among other observations, we could clearly see that the calcium carbonate layers of the shell began to lose structural integrity towards the end of the experiment. Overall, the survival of 75 mussels per treatment was logged throughout the 6 weeks, and all manipulated treatments showed a lower proportional survival compared to the current climate control setting. The lowest survival rate was seen in the climate change treatment, with only about 67% of mussels surviving. An additional 18 mussels per treatment were individually marked and measured in 3 dimensions on a weekly basis. The volume of an ellipsoid was used to approximate and calculate volumetric growth of the mussels, and we can see that seawater temperature and pH had opposite effects on the growth. The stimulating effect of an increased temperature was suppressed by the adverse effect of a lowered pH when combined in the climate change treatment. Next to these long-term observations, mussels from all treatments were used in weekly 3-hour incubations, and oxygen consumption was measured for the whole mussel or for the dissected shell with an intact biofilm in order to calculate respiration rates. Empty seawater incubations were used to correct for the background respiration going on. Also in parallel, nitrous oxide production was measured, but these results are not reported in this presentation. When looking at the top graph, the mussel respiration rate, we can see that as a proxy for metabolic rate, the respiration was highest in the climate change treatment
and fairly constant throughout all 6 weeks. Combined with the lowest survival results and previous results from muscle experiments, we could state that muscles struggle to sustain a higher energy demand in this stress environment. If we have a look at the lower graph, the respiration of the biofilm, we could see that this was highest in the ocean warming treatment, and the low oxygen consumption in acidified treatments decreased over time. And these findings also match other biofilm experiments that we did. Using a microprofiler and a retractable sensor, the oxygen microprofile of the biofilm on the mussel shells was measured on different locations. Based on aerobic respiration, we can see that the biofilm at the start, at week 0, and throughout the control treatment was on average 800 micrometers thick. And we can also see that both acidified treatments saw a reduction in thickness of the biofilm, with oxygen consumption only happening at 50 to 100 micrometers. Temperature seemed to enhance respiration, but might influence thickness and patchiness of the biofilm because some profiles didn't show any or have little consumption of oxygen. But we do hope that DNA profiling might help us interpret these results. Next to the results presented here, there's still ongoing analysis for the rest of the experiment, so I hope to present some more results later on. Thank you.
Good afternoon by now. I'm going to talk to you about fatty acid profiling of copepods to meet 3 certain SDGs. The first one, no hunger, climate action, and life below water. Let's first talk about microalgae. Why are they so important? They have the unique ability to synthesize certain omega-3 and omega-6 fatty acids, which are actually very important to all marine life and also to humans. They've been linked to cell development, to decreases in diseases, and even decreases in cancer. They're eaten by primary consumers, such as copepods, and they serve as energy vectors. So they make sure that fatty acids are available to all other living organisms in the marine trophic web. Now, what happens to these fatty acids when stressors are applied? Stressors such as elevated temperatures. We've done some experiments where we incubate copepods on different elevated temperatures, and then we have a look at their fatty acid profiles. What we saw is that they have the tendency to decrease over time. Now, this needs verification still because our first experiments didn't show any significant data, so we did some backup experiments, and unfortunately for you guys and for me, the data is coming in next week, so I cannot present if in fact we have some significant results here. Now, it can be argued that temperate benthic copepods are actually, they have a really high temperature range, so that means that maybe they're not really experiencing a lot of stress because they live in winter in 0-degree water and in summer in 24-degree water. So that's why we also want to look at tropical copepods. That's why I went to Ecuador and I redid this experiment as well. Also, the results of this will be coming in next week, so more on that next year. We also want to include pH in the coming up experiments, and we also want to do some analyses with chemical pollutants such as metals. Now, you might be wondering, what about no hunger, right? Because you know climate action, all right, that was in there. Life below water, obviously. Well, coastal communities, especially in development countries, they're actually really dependent on seafood for the uptake of their daily nutrition. So that means if we can indeed prove that fatty acids decrease due to stressors, then this might be a problem for people living there and depending on this food. Let's take Ecuador, for example. The largest city in Ecuador is Guayaquil, and Guayaquil has over 3 million people who are living there along the Guayas River estuary. There's a lot of banana agriculture, shrimp, aquaculture, a lot of big industries, so the estuary there has a lot of problems with stressors. Now, it is also home to one of the largest mangrove ecosystems on the Pacific coast of Latin America. And not just animals live there, but actually there are also local communities, fisheries communities, that are living in the mangroves in small villages, and they are dependent on these food sources. They're eating fish, they're also scraping oysters off of the mangrove trees, as you can see here, and they're collecting mangrove crabs. So all of these people, they're dependent on the nutritious value of all of these things in their ecosystem. So what we've been doing is we've been providing them with workshops as well, where we're actually telling them how their ecosystem works exactly, what kind of stressors are there, because a lot of them are not aware of climate change, ocean acidification. They're not aware that Guayaquil is causing a lot of huge stressors on their ecosystem, so they don't really know what's happening when they're not catching quite as much fish as they hope to catch. So yeah, we're hoping to continue this work. I'm sorry I couldn't present you with any results just yet. That's for the coming 5 years, let's say. But the take-home message here is that climate change and chemical pollutants are having an impact on the fatty acid profiles of the small animals and the microalgae at the base of the marine food web. And this can potentially also cause harmful impacts on the marine resources and the nutritional value of seafood or commercial seafood. So I would like to thank everyone who contributed here, and hopefully more next year. Thank you.
Okay, hello everyone. I'm a Tubimundo student, and I'm going to present the results of my master thesis. So I'm dealing with mercury in mangroves, and my study site is in the west coast of Peninsular Malaysia, and I'm looking at the impact of charcoal production on mercury pollution in mangroves, and particularly on the distribution of mercury in mangrove sediments and plant tissues. So what I collected for my mercury analysis was different plant tissues from Rhizophora apiculata and Rhizophora muconata. For instance, leaves in 4 different stages, roots and bark. And then I collected sediments both from the riverbank and from the inside of the forest. And additionally, I also did some analysis on animal samples, gastropods and cockles. And to analyze my samples for mercury, I used the direct mercury analyzer. And so for preparation of the samples, I washed the samples with distilled water, freeze-dried them, and dried them with mortar and pestle. Now, going directly to the results from the sediments, we found that the values of mercury in the sediments were quite close to the background level. Going to calculate the geoaccumulation index, that is a pollution index, we saw that all the values were below 1, and so they fall into the unpolluted category. But interestingly, what we saw also was that in station 2 and station 10, we found that there were charcoal pieces inside the sediments, and in these 2 particular locations, the concentration of mercury in the sediments was higher. So there might be accumulation of mercury in presence of charcoal pieces. Looking at the plant tissues, here we found very interesting results. For instance, as a general trend, we found no mercury in roots, and that means that there is no mercury uptake from the roots and then translocation to other parts of the plant. But we found mercury in leaves, and it accumulated over time with leaf stages. So in young leaves, there was less mercury than in senescent leaves. And this may be explained by atmospheric deposition of mercury on the leaves themselves. But strangely, we didn't find mercury inside the bark. And this is quite surprising because usually bark is used as an indicator of mercury pollution in the air. But in our case, we didn't find it. Then looking at animal tissues, in the cockles, the concentrations were all below the permissible limit for mercury in seafood. That's equal to 500 micrograms on kilo. And our values were around like 40 micrograms on kilo, so way below. And for the gastropods, we found that there was higher concentration than in plant and sediment tissues, plant tissues and sediments. But there was no homogeneity between sampling stations. And for instance, in 2 stations, the concentrations were lower. But we might say that there is some bioaccumulation of mercury inside the gastropods. And so to conclude, we can here's a summary of the results. And for future perspectives, we want to it could be interesting to to do some stable isotope analysis to see where the mercury is coming from and what is the speciation of mercury in the mangroves, because methyl mercury is the most dangerous for animals and humans. And so going to look if that is present in the cockles, for example, or even going to look at food chain, so going to look at fish, because carnivorous fish might have higher values of mercury than the gastropods or the cockles. And so thank you for your attention.
Okay, good afternoon, everyone. My name is Xiaoting Zheng from Laboratory of Aquaculture and Artemia Reference Center. Today, I'm so pleased to have a presentation here. My topic is evaluation of the anti-weevil activity of essential oil and their components. Weevil larvae is one of the numerous weevil oocysts and also one of the major pathogens of aquatic animals. Once they outbreak, you can see a phenomenon called bioluminescence here. Bioluminescence. Glowing blue water wash up on the beach. Is it beautiful? Actually, they can cause significant losses in aquaculture industry. So we need to find out a solution. Since the past few decades, essential oil and their components have been widely used for bactericidal and fungicidal. First, we need to know what is essential oil and essential component. Essential oil are complex mixture of secondary plant metabolites, and essential oil contain many essential components which are pure chemical composition. So today I got 34 kinds of essential oil from Leuven University. What we want to know is how anti-weevil of essential oil and their component. Will they act like killer, damage the weevil violently, or will they act like inhibitor, just block the colon sensing between weevil? So my hypothesis is essential oil and their component might have antimicrobial activity and colon sensing inhibition. My objective is to select the best candidate to control weevil in aquaculture. So I set up 3 experiments. Experiment 1, vapor phase mediated susceptibility. In this plate, I add weevil in the whole plate except H1 and H12. And in the middle of 4 well, I add essential oil on the top as volatility center. So according to the distance from the center, we can classify 15 categories like the table shows here. So here's the result. According to the pictures here, the well more close to the center, the well become more clear. Especially in C25, the last one plate, the whole plate are completely clean after 24 hours. Is it amazing? Yes, it is. And we can get 5 essential oil and 3 essential components, which can kill the bacteria while they vapor phase. So I'm thinking, what if I put essential into the weevil? What will happen? So I set up experiment 2, a bacteria growth assay. Here we can get 3 essential oil and 3 essential components, which can kill bacteria in the broth. So I thinking again, like most of the essential, they cannot inhibit the growth of bacteria. So maybe they might interfere the colon sensing between weevil as an inhibitor. So I set up experiment 3. So what is colon sensing in bacteria? Actually, it's a process of cell-to-cell communication. So here I got 4 essential oil which can interfere the communication between bacteria. So now we can have some take-home message here. From the experiment 1, vapor phase mediated susceptibility, we have 5 essential oil and 3 essential components, which can kill bacteria while they are vapor phase. From the experiment 2, bacterial growth inhibition, we got 3 essential oil and 3 essential oil components, which can kill bacteria in the broth together. So from the experiment 3, bioluminescence, we have 4 essential oil which can interfere the bacteria communication. Last but not least, we can choose the best 3 candidates here to control the weevil in aquaculture. So that's all my presentation. Thank you for your listening. I appreciate your attention.
Thank you all. It was an excellent session, I think. All presenters did very well. You can now vote for your top 3 presentations and submit your form later. We now have a next session of elevator pitches, 1-minute elevator pitches. So again, I would ask all the presenters to line up in front so that we can have one after the other. And the first one is Adede.
Good afternoon. Adede specializes in the detection of unexploded ordnance. And to this end, we actually use an array of geophysical techniques and various specialists. It's a bit hard to explain in 1 minute, so I would say if you have any questions about what we do or how we do it, please come and visit us at our booth. Thank you.
Hello, the blue cluster, as the name says, is a cluster of companies that are active in the blue economy. And basically we provide services to the companies. So we want to transfer knowledge because a lot is already known, so research is done by you, and we want to take that knowledge, the research results to the companies. And based on that knowledge, companies can think of innovations, so they can start to develop new products, new services, or optimize their activities. And we want companies to do it together with other companies, but also together with researchers like you. So collaboration is essential for the cluster. So we're active in 6 different domains, which you can see over there. So I would invite you, if you think your research results are relevant for industry, so you see some applications, so please contact us.
One minute is far too short to explain who is GXYZ, but I will try. We do geophysical and geotechnical survey. Most important in a company is the people, the equipment, and the vessels. That's the biggest one, 80 meter. Small timeline. We started 20 years ago as a topographic company, went through topography, inland waters, harbors, nearshore, through the offshore wind, and now active in offshore oil and gas as well. That's our mainly work in Europe, mainly the North Sea. We collect data with all kinds of geophysical techniques in a safe way. So no incidents and accidents, that's the goal. Certification, people are most important, field and office personnel, and our vessels, of course. Thank you.
I only have 1 minute, but I have two important messages to bring. The first is that over the past decade, we have seen the fields associated with marine construction and its effect on the marine environment grow, get more complex, and get more specialized. Every single one of our projects requires the tackling of a set of multidisciplinary issues, and therefore it is very important for Jan de Nul to meet the actors, the stakeholders, and the scientific specialists. And therefore we greatly support an event such as the Vlis Marine Science Day. And that is not only because they serve Brugse Zot afterwards, but it's something we really want to get behind. The second message for today, the second message is that Jan de Nul is hiring. We are looking for people, international and national talent, to join our specialized teams. So we have a booth set up here. Please join us. I thought the applause was for the beer. Sorry.
Good morning. Like any employer, ILVO needs employees. So the most important objective at ILVO is that you feel good in your job, that you feel free to express your opinion. And to achieve that, we have reduced the hierarchy, the rules and regulations and the structures as far as possible in order to create a dynamic environment where it is fun to work. So ILVO carries out a lot of applied research on marine ecosystems, on fisheries, on other uses of marine resources, and this work has an effect on our society. We need and we have a lot of different skills. You name it, we got it. So check out ILVO at Meet the Company. Check out our website for job offers, and we hope to meet you soon. Thank you.
Hello, I'm presenting the Operational Directorate Natural Environment of the Royal Belgian Institute of Natural Sciences. That's a mouthful. Most of you will know the institute as the Dino Museum in Brussels, but there is much, much more to say about it. So the OD Nature is a center of excellence in fundamental and applied research of biodiversity in ecosystems in support of the protection and sustainable management of natural environment. And that is only introduction to the slide that is way too busy, but that's what we are, very busy in many disciplines and fields. Both in marine, freshwater and terrestrial environments, but of course here it's about the marine part. Studies, monitoring, databases, major scientific instruments, and representation of the federal state in many international bodies are the tasks, and I've projected a range of fields of discipline because I knew I was not going to make it in 1 minute.
Hello, at Vlis there are different opportunities to get involved. If you want to be part of our team, please subscribe to our Vlisin newsletter or follow us on social media. Very soon we will launch more than 15 vacancies for different scientific profiles. But also as a student there are different opportunities. You can apply for a student job, you can write a thesis together with us or perform an internship. For all questions, please send an email to jobs@vlis.be or visit us at our booth. Thank you.
Since you're all here, I guess you're big science fans, and if you like a good beer too, then I have the perfect event for you. It's called Pint of Science, and Pint of Science is an annual science festival which takes place every May. And it brings together scientists in a local pub talking about the recent findings in the interesting world of science. For the first time, Pint of Science will visit Ostend on the 21st, 22nd and the 23rd of May in the CAP in Ostend, and it will be organized by Vlis. So let's come to our pub the end of May. Thank you.
Good morning. As far as we know, Tropimundo is the only master's program that actually brings the students to the field for an entire semester to study on tropical biodiversity and ecosystems. And we actually start our semester here in Europe in one of the partner institutes, bring them to then one of the ecosystems of their interest, which may be tropical rainforest or coral reefs in Malaysia, to then come back to Europe in another university as per the Erasmus Mundus regulation. We're also working together with a lot of associate partners that help us in providing internships and thesis opportunities to link the research to the people. And in that context, we are also proud to serve the Sustainable Development Goals by offsetting all the carbon of the mobility that we cause. Thank you very much.
Hi again. If you're interested in offshore wind energy or other forms of wet renewables, then I invite you to check out the poster on the REN initiative that groups 12 countries that are working together to resolve environmental effects of wind energy. You can find this poster when you go in the big hall immediately to your right. The thesis knowledge base that is an important asset here is an open repository of metadata supporting reports, relevant papers, videos, and other materials providing the current state of knowledge of the environmental effects of marine renewables. And you can see that it is addressed at a wide range of target groups. I'm sorry I had to look at my face twice. I invite you to come have a look, however, a third time in our info booth. Thank you very much. Thank you all. Before we break for lunch, we will have a group picture right here. So before you leave, gather in front here and we'll have a picture taken. Then we break for lunch and we reconvene at 1:30 sharp here in the auditorium.